Everyone. Welcome to part one of chapter 12, where we're going to be talking about something that is exceptionally important when it comes to development and disease and involves cells, and that is known as the cell cycle. It is the cycle of growth and division in a cell, allowing us to be multicellular, allowing us to develop complex limbs and organs and tissues. It's hard to imagine anything more important than this. The growth phase, what we call the interphase, and then the mitosis phase. And we're going to be covering both aspects of those things. It involves cell signaling and involves proteins and lots of things that we've talked about leading up to this chapter. Now, it's easy to understand that something like, for instance, cancer, which has at least 5 or 6, it's got 6 here, but I think it's now up to 10 different things that sort of have to happen in order for a full-blown tumor to arise. One of these things is sustained proliferation. One of the fundamental aspects of cancer is that it is inappropriate cell growth. That's the very first and foremost. And then whether or not it spreads in and invades other tissues. But you have to have all of these things. So right at 12 o'clock, you have to have inappropriate proliferation. And this is shown right here in this figure. This is kind of known as a Vogelgram, named after Bert Vogelstein at Johns Hopkins. In any case, what he did was he was studying tumors, and he would look at normal tissue, and then he would look at very, very early tissue that was on its way to becoming cancerous and more progressive and progressive and progressive. And what he found was that as you began to get more and more abnormal, you began to see certain genes being mutated. For instance, CDKN2A here is a cyclin-dependent kinase. This is directly involved in the cell cycle. KRAS is involved in proliferative signaling upstream. So what will happen is when this pathway, and this is part of a signaling pathway, gets activated, it will actually turn on genes like CDKN2A. We have TP53, which is actually a tumor suppressor, and that prevents a cell from going through the cell cycle. This one gets inactivated, and so forth and so forth. We see many, many genes involved in cell cycle and mitosis that are involved in development of things like breast cancer and pancreatic cancer and colon cancer, and the list goes on and on and on. But we can go the other direction. We need to understand cell cycle and mitosis in order to perhaps one day find a way to help folks who have lost their limbs in different circumstances in life, in this case, war. We know for a fact that in certain vertebrates that are very similar to humans, you can actually amputate a limb and it will grow back, not only back its, a stump on the end, but actually grow bones and cartilage and muscle and become a functional appendage again. We have tissues in our body that are capable of regenerating. Even neural cells, certain neural cells can regenerate. So we know the machinery is there, but for whatever reason in humans, that pathway has been turned off, but has yet been retained in certain amphibians and other animals. We know it's there. So it's a question of understanding mitosis, understanding what turns cells on and off and how they pattern and signal, and we can get back to here. And a little bit of hand waving for you, just to include this, that one of the key characteristics of something that is alive is its ability to reproduce. And so as multicellular organisms, yes, we can reproduce our whole organisms, but that depends on individual cells being able to reproduce. Reproduce literally tens of trillions of cells every day through this process of mitosis and cell division. There's another process called meiosis, and that's the creation of reproductive cells, which we're going to actually talk about in the next chapter. It's still a form of cell division, it's just that it has asymmetric genetic material at the end. Including this picture just to show you, help you envision the process of what we call cell division and mitosis. In interphase, we have the chromatin is relatively unpacked. These are picture of nuclei. And so the chromatin is unpacked like your clothes are unpacked in your closet. Think of your nucleus as like a closet where all your clothes are hanging and ready to be accessed. What happens though is when we start to proceed into cell division, we start packing that DNA, getting it into a very, very small package that we call chromosomes, which is a combination of DNA and protein. Those chromosomes are arranged, they are split. In this case, they should be split evenly, and they divide, and then we form new cells and unpack those clothes that have been copied. Okay. You can even sort of see it here. If you look here, this is sort of to scale. You can see that the DNA has been replicated here. There's a lot more of it here, say, than this picture, and it's starting to condense through here. So this is just starting to condense when we're duplicating those chromosomes, and then we really condense them when we get down to here. And so when we have a cell dividing like this fertilized egg, which is just split into two cells, this is this is what we call mitosis. Produces the possibility of an entire organism. If these two cells split, we'll actually have identical twins. But we don't just develop with cell mitosis. Certainly development is a very, very important aspect of mitosis, but we also grow over time and we repair over time. For instance, your bones regenerate top to bottom roughly about every 10, 15 years. That you repair the bones, you repair tissues in your body, your skin, you're constantly sloughing off cells, so you have to regenerate those, your digestive tract. It all involves mitosis. It is a part of what we call the cell cycle. The cell cycle is the process of preparing to divide, copying the DNA, checking to make sure we have all the DNA correctly copied and we have the cells for mitosis, and then entering mitosis. The act of mitosis itself is actually a relatively short phase of the cell cycle. It's getting ready for DNA replication. It's checking the DNA and getting ready for mitosis that takes the longest period of time. And that's called interphase. Some pictures here showing you examples. Reproduction, that is to say, creating progeny, growth and development, growing from a very relatively small individual to a full-sized, and repair, tissue renewal, constantly dividing cells. Your immune system is that way. Lots of rapidly dividing cell populations in your body. And so cell division that happens in our somatic system of our somatic division of our cells, we call that mitosis. It's a form of asexual reproduction because you're creating exact duplicate copies, or should be creating duplicate copies of the original parent cell. We create what we call daughter cells. On the other hand, meiosis that produces our reproductive cells, sperms and egg, are asymmetric. They are asymmetric. Yes, they produce cells, and they are cells from our parent cell, but they have asymmetric distribution of genetic material. That's one of the reasons why meiosis is considered a separate process. There's a lot of similarity there. They're not going to reinvent the wheel. When it comes to certain processes, they're not going to reinvent the wheel if it works. What they are going to do is they're going to adapt the wheel in order to get that asymmetric distribution of genetic material. But mitosis, like any other process, involves cell signaling, and that's why we did cell signaling first. Here what we have, and this is really, really cool, we've had a sperm that has fertilized an egg, and right at that point of fertilization, we get this wave of calcium spreading through the cell. It's really, really cool. It shows cell signaling almost in real time right here. This happens literally in a matter of seconds, that you can see this wave of calcium spread through and actually establishes polarity on the cell for division. But cell signaling is a very, very important, protein function is a very, very important process in mitosis. So I wanted to kind of keep us in the cell signaling mindset going into this chapter. Here's another really cool one. This just came out this year. It is showing protein signaling. They have a certain label that will glow when this protein is being modified. And look at the wave pattern. This resembles quantum fluid motion. That's super duper cool in my mind, showing you how protein modification occurs in response, again, to fertilization in this egg site. Eggs are among the largest cells known. In fact, the largest cell in nature right now is the yolk of an ostrich egg. It's one giant cell, the yolk. And so they study egg cells because they're easy to study. They're nice and big, and you can get tons of them. And you can see, and they can modify them with these fluorescent proteins. And you can see this quantum fluid behavior of protein modification in the cells. Super cool. One of the really big tasks in mitosis is copying and organizing the genetic material. So we need to kind of go over that a little bit. The total amount of DNA, and this would really, I mean, I can kind of waffle a little bit here. We usually think of nuclear DNA as our genome. We have mitochondrial DNA, and I should probably just go ahead and modify my language a little bit. So all of the DNA in a cell consists of what we call the genome, even though we don't normally talk about mitochondrial DNA in that context. Usually we're talking about nuclear DNA. So we have to copy all of that. In the case of a prokaryotic cell, it's a single DNA molecule that's circular. In the case of eukaryotic cells, it's a number of linear DNA molecules. And that linear DNA actually presents with a little bit of a problem when it comes to replication. When we get to DNA replication in a later chapter, one of the issues is what we call the telomeres at the ends of the DNA. It doesn't copy correctly, so it's a little bit of an issue. And that may be involved in aging, actually. But we have a combination of those linear DNA molecules in eukaryotes and the proteins associated with them, and they're called chromosomes. And the reason they're called chromosomes is because they were able to stain and see them, 100 plus years ago. And so these are chromosomes. It's the DNA and the proteins associated with that DNA. And in the case of humans, we have 23 pairs, a total of 46. 23 pairs. The only pair that is asymmetric are the X and the Y. And here they are depicted graphically, that we can actually identify chromosomes by their relative banding pattern and their size. So here's XY over here, which we have right here. This person is a male genetically, and we have an XX for a genetic female. But you can actually identify, we can still use chromosomes for diagnosis to this day. I should say is that a bacterial circular DNA is also a chromosome. It's not just linear DNA. So here's a prokaryotic bacterium, and here is a eukaryotic protist. We can see the linear chromosomes right here. Chromatin is the material that makes up a chromosome. So chromatin is the DNA and the protein, and a chromosome is a complete entity made up of chromatin, if you know what I mean. We also have DNA in our mitochondria, and DNA are found in chloroplasts as well. So here's a lovely picture of condensed chromatin. We can see the individual chromosomes inside that cell. And how we get those karyotypes that you saw earlier, where they literally take a picture of a cell in mitosis, and then they cut out the pictures of the chromosomes and they sort them. That used to be done by hand. Now they can do it. Now they can do it electronically. In any case, you can see that this DNA has been packaged and is condensed, and this cell is going to be getting ready to divide sometime soon. So chromatin condenses when a cell is getting ready for cell division, but that's after the DNA has been copied. So the DNA plus the structural proteins is chromatin, and one complete entity of chromatin is a chromosome. This chromosome has actually been copied. This is actually two chromosomes attached here at what's called a centromere. So this is actually not a normal chromosome. This is a pre-mitotic chromosome. A normal chromosome would only be one of these half of this molecule. So every eukaryotic species has a certain number of chromosomes. For humans, it's 46. For other animals and plants, it's a different number. But we are diploid. That is to say, we inherit one set of chromosomes from our mother and the other half from our father. That's in our somatic cells. Our reproductive cells, our gametes, have half as many. They are haploid. That is to say, they have half as many chromosomes. So a sperm and an egg in a human would have 23 chromosomes as opposed to 46. And the way that DNA is packaged is just nothing sort of miraculous. A given strand of DNA is 2 nanometers. That is 2 1/billionths of a meter. It would take 500 million strands of DNA side by side to make a meter. And every cell in your body with our 46 chromosomes, if we were to attach them all and make one linear molecule, would be longer than a yard. It would be about a meter. So hold your hands out about the length of 3 feet or so. That would be your length of your DNA in each and every cell. And so we take that strand and we have to package it. There's just no way to fit. It's like trying to get a genie in a bottle. We have to wrap it around the structural proteins. These are called histones, these little yellow balls. And then we create these nucleosomes of DNA and histones. And then we take those histones and package them and spiral them and spiral them and spiral them some more and some more. And finally, we get to the chromosome. And again, this is a pre-mitotic chromosome. This chromosome will literally split right down the middle right here, creating one here for one daughter cell and one here for another daughter cell. So we start with that 2 nanometer strand. We create an 11 nanometer nucleosome, then make a 30 nanometer fiber, and then fold that fiber again and again and again, and we get that condensed chromosome. Now here's a little bit of some tricky language, and again, a lot of language here in the beginning. We have to replicate that DNA before we condense the chromosomes. When we replicate the DNA and create that sort of double chromosome, if you will, each individual soon-to-be chromosome is called a chromatid. While it's attached, it's called a chromatid, okay? And there's a centromere that holds those 2 chromatids together. While they are attached, you can see the light purple one and the dark purple one, they are called chromatids. As soon as they separate, they become chromosomes again. A chromosome is an independent body of DNA and the structural proteins. So right now, this is one chromosome with 2 chromatids. As soon as they separate, they become 2 chromosomes. That's always a little tricky in the language here, and don't be confused. Cell biology is very much a language class. And so here is that chromosome, and here it's showing the sister chromatids with that centromere. There are some different scaffolding proteins that will assist the migration of the soon-to-be chromosomes to either end of the dividing cell. Later on, that centromere will serve an important role, like for instance in meiosis. When we're talking about meiosis, the centromere will serve additional roles as well. In binary fission, which is what occurs in asymmetric reproduction, particularly like to say in prokaryotes, we get that DNA replication and segregation and cytokinesis. It's not unlike what we see in mitosis, but we have to arrange the chromosomes in a way that the individual chromatids will separate to distal ends. And that's why the centromere becomes really, really important. Mitosis is one of the most mutational cellular events in biology. That even though mitosis is very, very, very faithful, it's very good at replicating cells, it is still a process that will lead to errors from time to time. That's one of the reasons why inflammation is one of the major risk, chronic inflammation is one of the major risk factors in cancer. If you have chronic inflammation of an organ or a tissue, you're at risk of having more cancer there. That's like, for instance, sunburn. Sunburn is inflammation of the skin, causes mitosis, and leads to the possibility of a mitotic error that could lead to cancer. In any case, mitosis is the organization of those chromosomes and then the division of those chromosomes into daughter chromatids that are now chromosomes into individual cells. So when they are together, they're chromosomes with chromatids. Here they are now individual chromosomes. I like this series of pictures a lot because it's showing you in the interphase. So this is they haven't duplicated their DNA yet in this cell. This is a one chromosome cell, and it's showing you the number of chromosomes that are present in that cell. When we go through the S phase of the cell cycle, we create this double chromatid. We actually double the amount of DNA in this cell during S phase. In fact, you can actually measure that. If we were to stain DNA in this cell with a fluorescent probe for DNA and then stain this cell, this cell would be twice as bright, or certainly much brighter than this one is, because there are more molecules fluorescing inside that cell. And then when we undergo mitosis, so we're chromatids here, and now we're individual chromosomes here. Now, cell division, when it comes to the actual splitting of the cells, is actually two things. One is mitosis, which is the genetic material being separated. And the second is what we call cytokinesis, which is the physical separation of the cytoplasm. And these are two separate events. Yes, they are meant to occur in sequence, but you can have cytokinesis without mitosis, and you can have mitosis without cytokinesis. It does happen. We see it in cancer cells. So they are separate events. And so this is the physical cytokinesis here that's occurring, and this is late cytokinesis. We also have, like I said, meiosis, and meiosis actually has two different cytokinetic events. We have a sorting of chromosomes, and we're getting to a haploid state in division 1, and then we're doing actual mitosis in division 2. So we have division 1 and division 2 in meiosis. And again, we're not reinventing the wheel of cell division. We're just adapting it for the purposes of asymmetric, non-identical genetic daughter cells. I'm going to be really, really careful in this next slide because I think if I had my way, I would just ditch the term interphase. I don't know why we even bother with it anymore, but it's still part of the curriculum, so we need to talk about it. We have the active divisional phase that we call the M phase. They call it the mitotic phase, but the truth is there's actually mitosis and cytokinesis. Why? Because mitosis is the segregation of chromosomes from the original double chromosome, and we have the physical cytokinesis, and that's shown right here at M. And then we have all of the other stuff, which is basically about growing proteins and dividing the DNA. So mitosis is right here. And then the rest of it, if this were 24 hours, mitosis would be only about 1 hour. The other 23 hours, shall we say, and that's not abnormal for a human cell. In an actively dividing human cell, somewhere between 18 and 24 or so hours. So only about 1 hour is actually that physical mitotic phase. The other 23 hours are what we call interphase, and there are subphases in that interphase. So about 90% of the cell cycle, that interphase can be is what we're doing is we're preparing to copy the DNA, we're copying the DNA, we're double checking the DNA, getting ready for the active physical division, and then once we're all set, we go into the physical division. And so here are the three subphases of interphase. I mean, like I said, I could ditch interphase. We don't need it. It really doesn't serve any real purpose. Okay? But in any case, we have what's called G1. That's because we can't really observe a whole lot going on here, but we know it's exceptionally busy. It is preparing for DNA synthesis. It is checking to make sure the cell is ready for DNA synthesis, and that's called G1. Sometimes they say G1, G0, but we'll get there a little bit later on. Then we have S phase, which stands for synthesis. We are copying the DNA during synthesis. We are synthesizing DNA. We have the second phase, the second gap phase, G2, which is through here, and then we have the mitosis phase. So G1, long time, and again, the cell is awaiting for signals here. Sometimes they say G0 because if a cell is not dividing, it will stay roughly in this area until the appropriate signal comes along to tell it to divide. So once a cell has been told to divide, we start proceeding to get ready to duplicate our DNA. We have a checkpoint here, make sure we're all ready to duplicate our DNA, and then we enter S phase, where we are actively synthesizing DNA. We double the DNA content. Once we're done copying the DNA, we check to make sure it's all been copied correctly. We package it, we can get ready to condense it, and then boom, we hit mitosis phase, and that's where we really start packaging and dividing the DNA. Cell is growing during all three phases, but the only time we're duplicating DNA is during that S phase, during that synthesis phase. We're checking the DNA, preparing for mitosis, synthesizing the mitosis proteins, and then we package the DNA, segregate it, and do cytokinesis during the mitosis phase. And that's shown here. We can hang up in G0. Cells that are not dividing, like for instance, your neurons are generally not dividing. They stay in an early phase of G1. They stay perpetually in interphase, and they can stay there for decades or lifetimes. Once the signal to proliferate has been received, it will start preparing to synthesize DNA. We'll get to this checkpoint. If we pass the checkpoint, we begin synthesizing the DNA. By the time we get to the end of S phase, we now have 2 copies of our DNA. We can see this visually with DNA staining. And then we enter the gap 2. This is another process of ensuring fidelity of the copy, getting the DNA ready to be divided, synthesizing mitosis proteins, and then we enter that active mitosis phase where we package the DNA, segregate the DNA, and then segregate the cells. G0, G1, like I mentioned, sometimes cells stay in that early phase of G1. So let's look at the M phase now. And there are several subphases of the M phase, and many of you have probably seen this before. We package the DNA, we break up the nucleus, we prepare the mitotic spindle and get those chromosomes hooked up. We line up the chromosomes along a midline, the metaphase plate. We segregate those chromatids into individual chromosomes and pull them to opposite poles of the cell. In telophase, what we do is we begin reforming the nuclei and we begin pinching the cytokinesis, if you will. We begin pinching the center line of the cell and segregating them into separate daughter cells. And this usually happens quite fast. It happens within, sometimes just in a little less than an hour. So remember the 3 major aspects of the cell division during the M phase are to condense and segregate the chromosomes. So those 2, so condense and segregate the chromosomes, and then pinch off and create 2 individual cell bodies, that cytokinesis. So we have 5 subphases of M. So this is all in the M phase. The first phase we have is the condensation of the DNA. So we're condensing those chromosomes, and we're starting to break down the nuclear membrane. You don't really do it until late in prophase. Okay? So prophase is where we're packing the clothes, getting ready to go on vacation. Over here on the left, that's interphase. This is what a normal cell would look like pre-mitotic. But once we enter mitosis, the first thing we'll see is the condensation of the genetic material. That is the hallmark of prophase. We still have a nucleus, but look at the difference between this one, and you can begin to see darker patches where the DNA has condensed. Prometaphase, and again, these are sort of slippery boundaries here. We break down the nuclear envelope and we begin organizing those chromosomes. They get linked up to what we call the mitotic spindle. They get linked up to these microtubules of the mitotic spindle, and they begin to get organized in a single line. When they are organized in that single line, that is the hallmark of metaphase. And so they're almost like a single file set of chromosomes here across the equator of the cell. That is metaphase. We call this a metaphase plate. A little hard to see here, but you can see that those chromosomes are lined up if you look closely. When the sister chromatids separate, that's anaphase. So you can see the difference between here and here is now that you can see space in between there. And so now those sister chromatids are now chromosomes, and they're moving to their respective poles of the cell. And then telophase is where the nuclear structure begins to reform. We see segregation of organelles and overt cytokinesis. So cytokinesis can actually begin in late anaphase. When these chromosomes are getting real close to the end, we can actually begin to see the formation of an actin ring at the equator. We can begin to see segregation of organelles. And then late in telophase, we have that overt pinching of the cell body into two daughter cell bodies. So that's where cytokinesis is occurring, late anaphase and in telophase. Right here as depicted by this red bar. Over here, like I said, that's interphase. We don't have condensation of chromatin yet. It's that condensation that really hallmarks prophase. So there's prophase. Prometaphase, we're shuffling those chromosomes, getting them lined up onto the equator, what we call the metaphase plate. So that's the hallmark of metaphase. Anaphase is when those chromatids have split, and now those chromosomes are migrating to the individual poles. And then finally, telophase and cytokinesis. To know these pictures, get to know the mitotic spindle. We're going to be talking about the mitotic spindle in the next part of our lecture in the next video. But we're going to be working on some of the organelles and so forth. And so get to know these phases. You'll be asked to recognize them and identify them from each other. So I just have some larger pictures here. I'm not going to redo this process. We are checking the DNA. We are synthesizing proteins for mitosis, duplicating the centrosomes and so forth. Okay, prophase, we're condensing the chromosomes. Yeah, you might see a mitotic spindle if you're lucky. Okay, we can really see that mitotic spindle in prometaphase where it's at the distal poles. Metaphase, the plate. Anaphase, telophase, cytokinesis, etc. And I think this is a super cool image where we can actually see all of the different subphases of mitosis. And let's start up here at the top. We can see the condensation, that's prophase, prometaphase, and then finally metaphase. Here we have metaphase, anaphase, and then telophase, cytokinesis going on in these two cells. Metaphase, anaphase, and cytokinesis telophase. Dramatic one right here. You can really see the cytokinesis going on right about there. So you can see all of these phases and the condensation of the chromatin, the breakdown of the nucleus. It's beautiful, beautiful image. Well, that's it for our lecture today. Thank you for your time, and I will see you in part 2 of Chapter 12.